DNA is the genetic data that dictates what we look like and how our body functions. However, only 2% of our DNA is actually responsible for coding the proteins that keep us alive. The remaining 98% of our DNA doesn't code for anything, which leads us to believe that they're just junk taking up space in our bodies. Though in the last few years, we found that these seemingly useless DNA are often underappreciated. So in this video, we will go over a brief history of junk DNA, what they are, and what we think they do in our bodies. Following the discovery of DNA in the 1950s, we have actually been slowly figuring out what it actually does. We found that it codes for amino acids, which is a crucial component for proteins. But we also found that amongst the DNA that code for those amino acids, there are long stretches of DNA that don't code for anything. Think of it like charms on a very long bracelet. The charms are the bits of DNA used in protein production, while the empty string is the non-coding DNA that doesn't make any proteins, which we refer to as junk DNA. Then, in 1990, we started the Human Genome Project to map out our entire genome and figure out what each part of DNA does. When we finally finished up the project in 2004, we found that only around 1.5 to 2% of our entire genome is responsible for coding protein. The rest are just sitting there doing nothing. So now what even are those non-coding bits of DNA? Think of our genome as a library, where the coding DNA is the books, while the non-coding DNA is the shelves, the catalog, librarians, and everything else that keeps the library in check. A big chunk of our junk DNA is made of repetitive sequences that can move around, called transposons. These chunks of genes will move around our DNA, causing mutations and ensuring a good genetic diversity. We also have introns, which are inserted in between the parts of our coding DNA that regulates how often a specific protein gets encoded. Satellite DNA is another non-coding DNA that makes up the main structure of centromeres and telomeres. During cell division, centromeres play an important part in pulling the cells apart after the DNA is replicated. Meanwhile, telomeres are at the tip of our DNA, protecting it from becoming tangled like the eglets in our shoelaces. Lastly, non-coding DNA can also be transcribed to make non-coding RNA, such as rRNA. These RNA molecules are used to make ribosomes, which are the protein factories of a cell, but the rRNA strands don't make the proteins themselves. And there we have it. Despite being commonly referred to as junk DNA, we need all 98% of them for our body to function properly. We are very lucky that only 2% of our DNA is responsible for encoding proteins, since when we get random mutations, it is far more likely for them to occur on non-coding DNA that doesn't affect us too much. These non-coding DNA help us regulate protein production, create more genetic diversity, and ensure that DNA replication goes smoothly. So in the end, they aren't really junk DNA, they are more so the hidden files in our massive catalog of DNA data. I hope that you all have learned something interesting today. Thank you for your time, and stay hydrated.